Okay. Good morning. Can you hear me okay? Yeah? Volume's good? My name's Professor Jarku, and I will be teaching you probably 3/4 of this course. The other quarter will be taught starting end of September, intermittently with me with Dr. Vicente Lotario. Okay? So, I teach pretty much this course at Georgetown University to the first year medical students. The lectures that you get here are pretty much the same lectures, slight variations, but not too much. So if you can do well in this course, my course, then you can handle medical school histology content, okay? Which I think a lot of you are here to find out. Can you handle the material? Now there is quite a bit of material, so you really need to keep up with it, okay? Study it as you go along. Don't wait, because you'll get overwhelmed. There's a lot of content. Having said that, let's see if this will advance. Maybe try this one. Seem to be having an issue. Okay, terrific, thank you. Okay, so texts. So here is the textbook, the histology textbook. That's number 1 up here, right? Shankara, which we have been using for many years, and it's updated periodically. This is a really good textbook. Many of the images that you will see in here, you will see in my PowerPoint presentations. Not every one. You know, I get slides from different locations, but many of them are from this textbook. This textbook you have access to for free online through George Mason University and also through Georgetown University. I think you've been told that already, right? So you don't need to purchase it. This is recommended, it's not required. And obviously since you can get it free, it's not required. If you do want to purchase it, if you like a hard copy, you certainly don't need to purchase the new version. You can probably go online through Amazon and get an edition that's a couple of editions old for a few dollars, right? And really things haven't changed that much. They color a few images. There might be a paragraph here or a paragraph there that's different. So really I would not spend the money on buying a new copy unless you really want it. I would do it online, Amazon. You'll get it really cheap, and then you'll have a hard copy if you want that. And then of course you don't need to have a copy of that. Also, I recommend Paulson. It has a lot of exam questions and board review questions in there, so that will be good to help you studying material as you go along, okay? Now, of course, we have the syllabus. The syllabus, which you can see online, has been posted, right? Gives you the dates and the lectures and the lecturer, okay? That's online. So you should look at that to see what the upcoming material will be if you so choose. You certainly don't need to, okay? And then I've also listed an atlas here for looking at many different pictures of some of the images that we're going to be having. So you may see from this syllabus that we have both lectures and lab sessions. So the lectures are basically a PowerPoint didactic presentation, right? And then the lab is also a PowerPoint presentation, but it's more informal, where you have an opportunity to ask me questions, right, about structures, because that's more identification, okay? And so we'll have both of those for every organ system that is listed in the syllabus. Okay, examinations, all right? There's 4 exams. Exams are on the dates specified, okay, which is here listed. So mid-October and like the second week, I think, of December. The dates are posted there. We can't change the dates, okay? There's 110 of you this year. If I change the date for one person, by the time we've gone through 110, we'll have had to have taken the exam yesterday, right? So we can't change the dates. Periodically, I get students asking me, well, Grandma's birthday is on the that particular birthday, on that particular exam day, or there's a particular religious holiday on that particular exam day, or my cousin is coming in from Europe that day and we want to go and party that day. It's like, sorry, okay? So look now at the dates, plan accordingly, okay? We will not be changing the dates. They are the dates. So you've been told, because I know somebody's going to come along and want me to make some sort of date accommodation. It's not going to happen. So the exams, the first set of exams, the midterm, consists of a written exam and a laboratory exam, a practical exam. So the written is worth 30% of the total, the end of year exam. The first lab exam is worth 15%. Gets you kind of used to what to expect in the lab exams. The second written is also worth 30%, and the second practical is worth a little more, 25%. And the second practical is also cumulative. In other words, it may have questions in there that you learned in the first half, okay? So that's the breakout of the exams, and that adds up to 100%, okay? So that's kind of the overview. Any questions so far on that orientation? And of course, if you have questions, we have 4 teaching assistants for you this year. 3 of them are going to be teaching Histo. They are your day-to-day go-to resource. They're here all the time, at least one of them will be. If you have questions for me, you can ask me questions at the end of the lecture. If you want to meet with me in private, if you feel embarrassed about some question, or for whatever reason you just don't feel comfortable speaking in front of other people, let me know at the end of the lecture so that then I can stay in my office up on the 3rd floor, right, just near where Dr. Bill Hahn's office is, and the same level where our teaching assistants are. I will make sure I stick around and stay there to talk to you, okay? If you don't ask me to stick around and have a office visit, then I will leave, because then I go back to Georgetown University, okay? So you have 2 opportunities to speak to me right after the lecture, or ask to meet with me after the lectures, and then we can meet in the office, okay? All right, and of course the TAs are here all the time. They've done this, they did all of this at Georgetown University. It's just pretty much the same material. They did very well, that's why they're here teaching, so they know it probably just as good as I do. Okay, so then you can ask them, and if they have any questions, they can email me, and also I believe on Blackboard there's a discussion board where you can also ask questions and get answers. Okay, so having said that, I am here to help you, okay? I'm here to help you do as well as you can in this course. Okay, the questions are at the same level, more or less, of the first year medical students. I'm not trying to be tricky in the questions. I'm not trying to make them too hard. I'm not trying to show you how smart I am, because I'm really not that smart, okay? What I'm trying to do is to help you. I've got nothing to prove to you, right? So the material that you'll get is here to help you as best as I can, for you to achieve the best that you can to move on to the next thing that you may have planned, okay? Having said that, there is a lot of material. You do need to keep up with it. One way to keep up with it is to have little study groups. Sometimes if you have 2 or 3 or 4 students, one of the students will present that particular lecture's content, and you can question each other. It's a way, question and answer, then you really kind of know what you know and what you don't know. And what you don't know is the most important thing, because then you go and learn it, okay? So keep up, and if you have part-time jobs, I think that's going to be problematic, because there is a lot of content, okay? You might fall behind. I know you've paid a lot of money for this course, for this certificate course, and there's a lot of content in it. So you don't want to waste that money, and you don't want to get a crummy grade because you didn't have the hours, couldn't spend the hours studying the material. So that's, of course, a personal decision for you, but just let me throw that out there for your consideration. Okay, having said all of that, the first lecture then is histologic techniques. So how do we look at tissues and organs in the body? Because basically we're going to survey the entire body, every tissue, every organ of the entire body. By the time you're finished with this histology, because you will know so much about your own body and how it works, you know, you'll be, I think, very pleased with your amount of knowledge from this particular course. So what do you have to know? Our objectives. We're going to look at the different types of microscopes used for histological examination, and we're going to look at the different types of structures we can observe and the level of resolution that we can observe with these different microscopes. And you need to know what structures can be observed with different types of microscopes, okay? As well as a bunch of other things that I tell you. But they're the overall learning objectives. So the first microscope that we may use is a light microscope. I think most of you are familiar with a light microscope. Basically, you have a light source that goes through various glass lenses that magnifies the image through a microscope slide, and then you observe it with the eye, and of course you can take pictures. So with a microscope, light microscope, you can resolve structures down to about 0.2 microns. And here's a formula that's used for the resolution of a microscope. The wavelength, which will be the wavelength of light, times a constant here divided by the numerical aperture. So numerical aperture is the ability to resolve two fine specimens. So here, my fingers are two specimens. You can resolve each one, right? As you move the fingers together, eventually there comes a point where you can't distinguish one from the other. Okay, so that's resolution. Now you can resolve two structures. Now you can't. There's just one structure. Okay, that's resolution. So with a light microscope, you can magnify things maybe 1,500 times. Obviously, some microscopes are better than others, but a good microscope will let you magnify it 1,500. Another type of microscope is an electron microscope, a transmission electron microscope. In this microscope, instead of glass lenses to focus the light, we have electromagnets that focus a beam of electrons. So we shoot a beam of electrons down through this electron microscope, and the electromagnets focus that beam of electrons through an image, and we can resolve things much smaller. So resolution with a transmission electron microscope is maybe 1.5 nanometers, and we can see things maybe about 500,000 times, 500,000 times magnified. Okay, so obviously electron microscope, we can magnify things much better. There are different electron microscopes. Some are better than others. Some specialty ones can even go much higher than this, but the typical one that most universities and research labs will have do about 500,000. And then we also have a scanning electron microscope, which is also using electrons, but basically it scatters electrons on the surface of a structure, and so we can see fine detail of that structure. Basically, scanning electron microscopes allow us to observe surface topography, okay? Not inside the cell, but what's on the surface of the cell or the structure. And scanning electron microscopes, we can see things maybe down to 2 nanometers and magnified 100,000 times. They're the three basic types of microscopes. Now, there are other types of microscopes that have been invented and used, and there are variations on some of those microscopes. Like with light microscopy, you can use fluorescence light, where you stain the slide with a fluorescent dye, so it will show specific structures. So there are variations, but those three, the light microscope, transmission electron microscope, and scanning electron microscope, are the three microscopes that we will be using and discussing to look at structures in this course. So here is a picture of red blood cells with the light microscope, okay, LM, light microscope. with the transmission electron microscope, we can cut right through a red blood cell, or 3 of them in fact, and now we can see inside the red blood cell, which actually isn't much to see in a red blood cell, which we'll discuss later on. Or with a scanning electron microscope looking at the surface topography, we can see that the red blood cells have this donut type indented appearance. Okay? So scanning electron microscope is very useful to get kind of an overall view of a structure of a cell that we may be looking at. Okay, so then going back to our histological sections, here is a light microscopic section actually of the kidney. We can see proximal convoluted tubules, distal, and a collecting duct. Now you can see nuclei, and you can see the border between 2 cells. You really can't make out too much within the cytoplasm of the cell, and you can make out some fuzzy microvilli on the surface. If we use a transmission electron microscope of the same tubules, you can see much finer detail. We can see the nuclei. Now we're picking up mitochondria in the cytoplasm, as well as a few other organelles. And we can make out microvilli much finer. Okay, so we're getting a much better level of resolution with a transmission electron microscope. With the scanning electron microscope, now we're looking at the surface topography. Again, this is the proximal convoluted tubule of a kidney, which really you don't need to concern yourself with. The point here is that you can see the surface topography. You can see what these microvilli look like from the outside looking down. So how do we prepare tissue for microscopy? So for light microscopy, we take a piece of tissue and firstly we fix it. So basically we use formalin, which is a fixative. It cross-links a large number of proteins to hold everything together. And then we dehydrate it. So basically we pass that tissue through a graded series of ethanols, starting out with like 30% ethanol, 30% ethanol water, 50%, 70%, eventually 100%, which removes all the water. And then we have the tissue dehydrated, and then that ethanol impregnated tissue is then impregnated with xylene, which is another organic compound which is actually miscible, in other words dissolvable with paraffin. Because then what we do is we take that xylene impregnated tissue and we put it into a block of molten paraffin wax, basically candle wax, okay? And then you let it harden. So now you've got this tissue impregnated with candle wax, and then we cut that tissue, that block of paraffin embedded tissue, on a microtome. So when you turn this handle, this goes up and down, this stage, and there's a steel knife here, and you can see here this sliver of tissue that's been sliced off the end. And then you take that sliver of paraffin wax, which has the paraffin embedded tissue in the middle of it, and you put that on a microscope slide, and then you heat the microscope slide, it dissolves, and then it adheres to the glass microscope slide. Then you let it cool, and then you pass it through a series of solutions, ethanol, starting out with xylene, then ethanol, and then eventually dyes. And these dyes allow us to see different structures within the cells and tissues. Okay, so that's light microscopy. Here's a piece of kidney that has been stained and prepared for light microscopy. And then, of course, we look at it under the light microscope, and we can take pictures with the camera or just look at structures. So here are the common stains for light microscopy that you may see throughout this course. I might mention that this particular tissue is stained with H&E. Hematoxylin and eosin is the go-to standard stain for pretty much everything first pass, okay? And the hematoxylin, which is like a purple stain, will stain the nucleus and nucleic acids, ribosomes, things like that. The eosin is a counter stain for pretty much everything that's cytoplasmic. So you get a purple and a kind of a, I guess it's a pinky orange eosin color counter stain. And that lets you see pretty clearly many of the structures within a cell at the light microscopic level. Then we have other more selective slides. There's the periodic acid Schiff stain, PAS stain, and this stains sugar residues. So sugars within, there are many sugars, compound sugars, attached to proteins like glycoproteins or glycogen. You'll see in the liver. This will stain disaccharide or saccharide residues, sugar residues. So we'll see that periodically. There's also aldehyde fusion, which will stain elastic fibers. So when we talk about connective tissue, I may discuss aldehyde fusion staining of elastic fibers. We also have an aursine stain, which will stain collagen fibers and sometimes used as a cytoplasmic counterstain. And then we also have silver-based stains, arginophilic. These will stain reticular fibers, which is a type of collagen fiber, and also the Golgi apparatus for some weird reason. So these 5 stains are the common stains that we may mention a particular light microscopic tissue has been stained with for observation. So this is an H&E stained section of the, looks like the respiratory epithelium of the respiratory tract. You can see nuclei and cytoplasm, and here these goblet cells didn't stain up much at all because they're filled with more of a mucinous material which doesn't pick up the H&E stain. Here's another H&E stained slide, looks like it. So the hematoxylin stain the nuclei, eosin is the counter stain of the cytoplasmic material. So this is typical of what we might see. Here's another H&E stain of the intestine. So here we can see the brush border stains up nicely. That's basically microvilli. B looks like a venule that's opened up. C is the lamina propria, which is a layer of connective tissue. D is showing you some of the cells of the intestinal epithelium. And E is the basement membrane upon which those cells rest. And F are goblet cells that are stained in this particular epithelium. So here's a picture now of the epididymis showing the Golgi apparatus that's stained with a silver-based stain. So but in particular, the Golgi stains very prominently in the epididymis. And so this would be a silver-based stain. Should be more black than red. I think that's just an artifact of the way the slide was reproduced. But these are the Golgi, stained with a silver-based stain. So now, whenever you're looking at a section through tissue, you have to keep in mind that you're looking at a single plane through, you're looking at a two-dimensional section, one plane through that tissue. So you could have a tube like this that's sliced at various locations, and you may see something like this and this and this and this, right? And you may think, well, this is a bunch of different tubes, but no, in fact, this is the one tube that has been sliced at different orientations. That might be a perfect cross-section, oblique section, longitudinal section. Keep that in mind when you're observing different structures. Again, here, look at this tube that's highly folded, like this piece of spaghetti. Here's our slice through the tissue, and you see all these different cross-sections. You might think there's multiple tubular structures, but in fact it's the one tube. Same thing here, cross-section, cross-section, oblique section. So keep that in mind, keep orientation in mind. Okay, so transmission electron microscopy. Now we're looking at things that are much higher magnified. So here we use different fixatives. We use formalin, glutaraldehyde, which again cross-link many of the intracellular structures, and we also use osmium tetroxide, which will stain up some of the membranes. Okay, nasty stuff, but it's very useful for, it's very volatile, but very useful for staining membranes in cells. And after we fix these tissues, again, we dehydrate them through ethanol, and then we embed them. Here's our little tissue. This is a little, like a little cup here, into which we've put our little piece of tissue that's been fixed, and we then put in there EPON. So EPON, you may be familiar with from the hardware store. So sometimes when you want to glue something really rock hard, you know, there's 5-minute EPON. So there's 2 tubes, right? And then you mix these 2 tubes of stuff together, okay, in 5 minutes or sometimes 30 minutes, it will turn rock hard, okay? It's kind of acts as a glue. It's the same stuff, okay, EPON. So then this EPON, which is liquid, then impregnates into the tissue, and then we put it in an oven overnight, and then when we do that, it turns rock hard, like so. So once that tissue is turned rock hard, we can pull it out of that little cup that we had prepared, plastic cup, and then we trim the very edge of that now rock hard EPON in preparation for putting it in a microtome, as shown here. So here's this little piece of tissue here, and this is a boat filled with water, and this is a stage that goes up and down, and it cuts a super thin slice on a diamond knife. This is a diamond tipped knife, which gives you a ribbon of tissue, floats out onto the water, and then you pick it up on this gold grid. So here's our ribbon of tissues that have been cut with this diamond knife, and the color of this ribbon, the color of these little sections, tells us the thickness of the sections because they're birefringent. So basically you get the different colors of the rainbow reflecting off these different sections, and those different colors tell us the thickness of those sections. The thinner, the better, obviously, for the electron microscope. So then we can, because these are floating on water, we can pick them up on a little grid like this. Many of them are copper or gold coated, okay, and they've got holes in them. And then we can put these little grids, well actually firstly we stain them, we put them on this little droplet here, stained with lead citrate and uranyl acetate, okay. The uranyl acetate's mildly radioactive. If you put a Geiger counter up to it, you'll hear it buzz, but as long as you don't get it on your hands, it's okay. It's not really radioactive, just mildly. So then you stain our ultra-thin section on the grid with lead citrate and uranyl acetate, which will stain up membranous structures. And so then this is what we get here for our transmission electron microscope. And then we put it in our electron microscope. We're shooting a bunch of electrons down through this structure, which has electromagnets which are focusing the beam of electrons, and is projected onto a fluorescent screen down here, which where we can see the magnified image, and then we can take a picture of it. And we'll see something like this. Again, this proximal convoluted tubule of the kidney. We can see now structure within the nucleus. We can see mitochondria and more detail of the microvilli. Okay, another type of microscope, another type of imaging that we can use is freeze fracture, where we take a piece of tissue, we freeze it, and then we whack it. And then what that does, what's that used for, is for looking at the structure of the plasma membrane. So the plasma membrane, which we will learn more about on Wednesday, is a lipid bilayer with proteins impregnated through it and around it. So the E face, so if we split this plasma membrane in two, the external face is the face of the plasma membrane touching the exterior. That's the E face, external face. So the the inner aspect of this external face, you can see these knobs here, which are proteins that have been, like here, projected through the lipid bilayer. Or maybe we cracked this transmembrane protein right across. The P face is the protoplasmic face. That's the other lipid bilayer which makes contact with the cytoplasm. So again, in here we can see protoplasmic face proteins that are embedded within this lipid bilayer. So that just helps us look at the surface topography, if you like, of certain proteins that may be embedded within the plasma membrane, the E face and the P face. So here you can see P face and E face, you can see these proteins that are embedded in these lipid bilayers of both of these faces shown here. Okay, the other third major type of a microscopy that we can use to observe cells and tissues is scanning electron microscopy, which is used more for surface structures. Again, for scanning electron microscopy, we have to fix the tissue with formalin, just like we did for light microscopy. We dehydrate it, in this case with ethanol, and then we dry the structure. We get rid of all the ethanol, so now what's left is just the proteinaceous material and nuclear material that was in the cell itself, and then we put it in what is called a sputter coating unit. So basically, a dried structure is sitting up here, and a mist of gold is sputtered over the surface of the structure. Okay, so we're covering the surface. So do you guys know the James Bond movie Goldfinger, right? So you remember how Goldfinger coated his girlfriend with gold, right? That's what happens here. Okay, but we're looking at cells and tissues. We're not looking at a whole person, but it's the same concept. The entire surface is covered with gold, sometimes with carbon, and like here's a bug on a stage here that's been covered with a very thin layer of gold, and then we put it in this scanning electron microscope, as shown here, okay? And we can see structures like this. So we can see the surface of a red blood cell. Okay? Surface of a red blood cell. We're not looking inside, we're looking at the surface of the cells. So here we can see the surface topography of the glomerulus of the kidney, which we will discuss. I think that's actually the very last lecture of the course. Complicated structure, but it's good to see the surface topography of structures because it really gives us a much better understanding of the cells that we can see that we've seen in cross-section. Okay, so in combination of scanning and either light or electron microscopy, we can get a much better appreciation of the structure of a particular tissue and the organization. You can see this is obviously different to that, so this is a different cell type to this component of the glomerulus. Or we can do something like this. So we know what this is, right? It's an ant, okay? And we can see the very fine detail, like look at these hairs on the surface of the ant. So here's his head, antenna, thorax, abdomen, and 6 legs, okay? So we can see very, very fine detail of the surface of the ant. We can appreciate the detail of that ant now much more so than if we were looking at a cross-section through it. We wouldn't really get the same appreciation of the various structures. So what is this? Spider. Nasty looking beast, isn't it? Yeah, it's like, what's that spider in Harry Potter? You know, there's one of those Harry Potter movies where they go into that tunnel and there's a chase, and it looks like that spider from Harry Potter. But again, you can see it's got like 6 eyes, and it's got hair all over its legs and structures like that. So it's, you can really appreciate the fine detail of what these small organisms or cells and tissues look like with scanning electron microscopy. Okay, well that went pretty quickly. So this is actually not unusual. So the lectures, we allocate an hour. Sometimes they go longer than an hour. Sometimes they go shorter than an hour. Usually if people ask me questions, that kind of stretches it out some. Today's went shorter, but that's fine. Okay, and quite often what will happen is, today we have 3-hour block, and we could have a break from 12 to 1 for lunch and then come back at 1. But in past years, usually what happens is the students would prefer to take a 5-minute break, and then we just go through all 3 lectures, and then you're done for the afternoon. Okay, so that makes it a lot easier on most of you. Then you have time to do stuff, other stuff. So unless you have a particular concern about that, then I will ask you every day, is that okay to go on to the next lecture or lab? And that way we get through it. So at the end of all of my lectures, I have a practice exam question, okay? So this would be typical, perhaps, of what you might see on the exam. I won't be this one, but it might be similar style. So a researcher suspects defective mitochondria may be responsible for a patient's condition. What type of microscope would be best to examine the mitochondria? The operative word here is best to examine the mitochondria. Would we use a light microscope, a scanning electron microscope, freeze fracture, electron microscope, or fluorescence, where we stain the cells with some fluorescent dye? Okay, I'll give you guys a minute or two to think about an answer. Okay, think about the level of resolution of structures that we discussed for the light microscope, the electron microscope, and the scanning electron microscope, as well as what freeze fracture was used for. And then the answer would be the electron microscope. Okay, so why is it the electron microscope? Well, the light microscope, sometimes you can make out fuzzy stuff in the cells, but you really can't resolve mitochondria very well. The scanning electron microscope we wouldn't use because that's looking at the outside of the cell. What the mitochondria are inside of the cell, right? So you wouldn't see anything with a scanning electron microscope. Freeze fracture was just for the membranes, the plasma membrane, so we're not going to see mitochondria there. The electron microscope is our best choice because we're looking at the intracellular structures with higher resolution, okay? And I showed you numerous pictures that had mitochondria in them with the electron microscope. Fluorescence, you know, that's a possibility if you had some specific stain for mitochondria. But again, this is at the light microscopic level. So for the same reasons that we wouldn't use A, we probably wouldn't use E if we had the choice of the electron microscope, okay? So that's kind of how you would, yes? Because you scatter electrons on the surface of the structure, and I guess the electrons bounce off onto some apparatus that captures the scattering of the electrons, which then is captured as an image. And I guess by practice, they tried different structures. They also use carbon sometimes, and there's probably other stuff as well. And they've just found that gold is the best, a fine mist. And also gold, you can make a fine mist of it. You've got to be able to melt and make a mist of some sort of metal or carbon that's going to coat evenly over your structure. Some compounds, probably, if you zap them, they would break up, right? With some organic, you couldn't use organic compounds because it would just break up into different pieces. It would become denatured. But gold, it's just a very uniform one element, so it's not going to break up, and it can be mystified, so it can then cover the structure. Another question. This one. Benefit of freeze fracture over electron microscope. Freeze fracture is pretty much exclusively used for looking at membranous structures. So you can see the membrane of cells with the electron microscope. You're looking at kind of different things. You're looking at a cross-section with the electron microscope. You will pick up some of the transmembrane and accessory proteins with the electron microscope, perhaps. Freeze fracture is really, it's almost like a scanning image with the freeze fracture because you can see the bumps of the proteins embedded in the different leaflets of the plasma membrane. So it's just another way of appreciating that the lipid bilayer is not just a lipid bilayer, it's got these proteins embedded in it, which is very important because when we do membranes on Wednesday, you'll see that those proteins act as pores, transmembrane pores, and accessory proteins for signaling. Okay, so they're very important. Was there another question? Yes. For the scanning electron microscope, is at 360. It can be. You can rotate the stage that has the image in there. You take a picture and that's static, but then you can rotate the structure, whatever's been coded, and get a different view. So you can get pretty much a 360 view if you can rotate it all the way around. Other questions? Okay, so let's take a 5-minute break, and then we will go on to looking at epithelium and glands, okay? Yeah, so like a 1:15, let's come back, and then we'll start the next topic. Good. Okay, so now the next topic is epithelium and glands. Okay, epithelium and glands. So you can see down the bottom here, a tissue is an aggregation of specialized cells that do different functions. There are four main categories of tissues. Epithelial tissue, which is what we'll be discussing today, right? Then connective tissue, there's a bunch of lectures that I'll give you on that. Muscle tissue, Dr. Nataria will be giving you that, and also nervous tissue. So they're the four main basic tissue types, okay? Epithelium, connective tissue, muscle, and nervous tissue. So we're going to be talking about epithelium, which is used to make up glands for the most part. So here are some objectives for you. Know the four basic tissue types, I just listed them. Be able to classify epithelium. You will be examined on classification of epithelium. Know the difference between an exocrine and an endocrine gland. Be able to classify a gland based upon tubular and secretory components. We're going to go over that in some detail. Know the different modes of secretion. We're going to discuss that. And know the different types of cell surface specializations and distinguishing features like microvilli, cilia, things like that, which we will see some of today, more of on Wednesday. Okay, so here's our four basic tissue types. Nervous tissue, okay, long processes. I think we kind of have a pretty good idea what a nerve looks like. Okay, really there isn't any extracellular matrix material surrounding nervous cells, and the function is obviously transmission of nervous impulses. Epithelia, which we will be discussing in detail today, these epithelial cells can be aggregated in different orientations, in different ways, but typically we have a bunch of polyhedral cells stuck together. And there's a little bit of extracellular matrix, usually at the basal aspect of an epithelial sheet, and these are widespread. So epithelia line the surfaces of pretty much all our body cavities as well as the external surface, and they line pretty much most of the glands within the body. And this is what we'll be focusing on today. Then of course we have muscle cells. These are contractile cells. There's three types of muscle cells. Okay, there's cardiac, smooth, and skeletal muscle. A little bit of extracellular matrix, and of course their function is locomotion, movement. Connective tissue, there's different types of connective tissue cells and extracellular material that helps to stick all of this stuff together, okay? And of course there's quite a bit of extracellular matrix, and their role is support and protection and adhesion, helping to stick different cells and tissues together. So there's our four basic tissue types. Here's some characteristics of epithelial cells that we're going to be discussing today. Pretty much all the surfaces of the body, excluding the joints. Okay, so joints, here I'm talking about articulating joints like the cartilage in the elbow and the knee and things like that. They don't have epithelial cells because where the joints articulate, the cells would be abraded off. Okay, so you don't have epithelia covering cartilage surfaces of articulating joints. We'll discuss that in more detail when I do bone lectures, cartilage and bone. But otherwise, pretty much all the rest of the body has some sort of epithelial lining. It also covers the exterior of the body. The skin is a type of epithelial sheet, okay? Epithelia line most of the passages of the body, so they line the digestive tract, they line the respiratory tract, the lungs, alveoli, and so on, and the urogenital system, okay? So the reproductive organs and also the excretory organs, kidneys, ureters, bladder, urethra, and so on. Epithelia also line various cavities. So the coelomic cavity, okay, where many of your abdominal organs reside, and serous cavities. So the cavity that lines the lungs, around the lungs, the pericardial cavity, the cavity that houses the heart. I mentioned the peritoneal abdominal cavity. Okay, these are all lined by epithelia, and they're given different names. So Mesothelium is often used for the epithelial lining of the abdominal cavity, whereas if we're talking about blood vessels, which are lined by endothelial cells, we're talking about epithelial cells that are described as endothelial cells. But they're all endothelial cells, whether it's mesothelial or endothelial, they're all epithelial. Epithelial cells also make up the secretory cells of many glands, and we're going to look at that in terms of how do we classify glands, how complex they are. Now, epithelial cells, they all reside somewhere on a basement membrane or a basal lamina. So a basement membrane is the term we use for a structure that we can see at the light microscopic level. Basal lamina is the term we use for a higher magnification of that same structure at the electron microscopic level. And we'll cover all of that again. Typically, the basement membrane can be PAS positive, okay? Remember we talked about those different dyes that we use to look at different structures. PAS positive at the light microscopic level. And the lamina propria we can see, the basal lamina we can see at the electron microscopic level. And in that basal lamina there are different types of proteins, collagens, some glycoproteins as discussed here. Now epithelial tissue does not have any blood vessels in it, okay? If it did, it would be a different type of, another layer of epithelial cells. So a one particular epithelial tissue will have no blood vessels in it. So that means that the epithelial cells have to get their nutrients by diffusion, okay, which is rate limiting. So diffusion is rate limiting, so you can only have a certain thickness of epithelium, beyond which diffusion is too slow and then those cells would die. So diffusion is rate limiting because epithelial tissue does not have any blood vessels. Epithelial cells also show polarity, okay? The apical surface of a cell might have microvilli. The basal surface of a cell will be residing on a basement membrane, and the lateral surfaces of the cell will be between two adjacent cells. So apical, basal, lateral, okay? So that's polarity, showing polarity as in north-south, if you like, or orientation. Sometimes we see keratin filaments, particularly attached to desmosomes, which are a type of intercellular structure that bonds cells together. Okay, then we can see it in cells and also attached to desmosomes. And these are very important for holding cells together to form an epithelial sheet. Epithelial cells have a good capacity for renewal and regeneration, okay? They're continually undergoing replacement, if you like, because they get worn out. The skin is a good example. The cells of your skin are constantly being shed and replaced with new cells. Epithelial cells can be derived from 3 different germ layers, so this is really embryology: ectoderm, mesoderm, and endoderm. And then, of course, these epithelial cells have a plethora of functions. Protection, such as the skin, lubrication, secretion, digestion, absorption, transport, sensory reception, reproduction. All of these things we will see as we discuss the various tissues and organs as we go along through this course. Okay, so surface epithelia. Okay, these are classified, epithelial sheets classified based upon the number of cells in the epithelium. Is it one layer of cells? If it's one layer, it's called simple. If there's multiple layers of cells, it's called stratified. And the height and the shape of the cell. So if it's flat, it's squamous. If it's square-shaped, it's cuboidal. If it's elongated, okay, it's columnar. So here we can see all of these. So the pink cells are the epithelial cells. And in all 3 cases, there's one layer of cells, which is then adjacent a basement membrane, which is this kind of purpley structure here. So because there's one layer of cells, we say this layer, these tissues are simple. Okay, simple means one single layer. If these cells are squamous or flat, obviously it's simple squamous. If these cells are more or less square-shaped, it's simple cuboidal. And if these cells are elongated, it's simple columnar. Okay, simple because in each of the three cases, the cells are just one layer thick, and the shape of the cell, squamous, cuboidal, columnar, is determined by the shape of the cell, the description of the cell. So that's pretty straightforward. And in all three cases, each of those layers of cells is on a basement membrane, and note there's no blood vessels anywhere to be seen within those epithelial cells. Remember, epithelia are avascular. So here are some examples. So here's a blood vessel, and at the very perimeter here, these are the nuclei in the cytoplasm. So this would be simple squamous. These endothelial cells lining this blood vessel would be classified as simple squamous. Simple because there's just one layer of cells resting on a basement membrane. Squamous because the shape is elongated. These cells are simple cuboidal. You can see they're more or less cuboidal. Okay, obviously they're not going to be perfectly cuboidal. You know, you've got to use a little bit of discretion here. Pretty much cuboidal, and there's a single layer of cells like here. So it's simple because it's a single layer and the shape is cuboidal. So this would be simple cuboidal for classification purposes. This is simple columnar. Again, there's one layer of cells. We're looking at the nuclei here and the cytoplasm. So one layer of epithelial cells, and the basement membrane would be right here at the bottom here. So this would be simple because there's one layer of cells, columnar because the cells are columnar shaped. Okay? Then things get more complicated. So here are three examples of different tissues that have their own name. So their own classification. Here we have stratified squamous epithelium. So stratified refers to the fact that there are multiple layers of cells with no intervening basement membrane. So here's just the one basement membrane. Here's one layer of cells, 2, 3, 4, 5, 6. In this particular case, there's 6 layers of cells with no intervening basement membrane, so we call that a stratified epithelium, like the skin. And we say this is stratified squamous because we classify stratified epithelium based upon the uppermost layer of cells. So this uppermost layer of cells, you can see, are elongated, so they are squamous, right? This more basally located layer of cells, they are, in fact, columnar. But classification is not based upon the basal layer of cells, it's based upon the uppermost layer of cells. So it doesn't matter what's going on down here. This uppermost layer of cells is squamous, so this is stratified squamous epithelium. So that's how we would classify this epithelium, stratified squamous. Then we have a relatively unique example of transitional epithelium, which we see in the bladder. Because the bladder distends when it's full of urine, or collapses when it isn't full of urine, the shape of the cells lining the bladder changes. Okay? They can be stretched out, that uppermost layer where the arrow is, or they can be compressed more and they become more cuboidal. So the shape of this uppermost layer of cells is transitional. It can change depending upon what's going on in the bladder. So we say then that this epithelium is transitional epithelium, and it's pretty much unique to the bladder and just a few locations where some cells, some tissues are leading into the bladder. Okay? That's a unique example. And we will discuss transitional epithelium in greater detail when we do bladder. Okay? And then the third relatively unique classification of epithelium is pseudo-stratified epithelium. Pseudo meaning false, falsely stratified. So this epithelium appears falsely stratified because we can see nuclei at different levels within this epithelium. Some are right down here, some are up here. But it turns out if you take an electron microscopic section through this tissue, every one of these cells makes contact with the basement membrane. Okay? Every one of them. Some of them pinch out, but still some of the cytoplasm makes contact with the basement membrane. So then, because every single cell makes contact with the basement membrane, they're not stratified, in fact. They appear stratified, but they're falsely stratified. So then we call this pseudo-stratified epithelium, falsely stratified epithelium. So we see this in a few locations. We see it in the respiratory epithelium of the pulmonary system, and we see it in the epididymis of the male reproductive system. Okay? You don't see it a lot. Pseudo-stratified epithelium. And in this particular case, they're saying that it's ciliated because the surface of these cells, and in this case, it would be the respiratory epithelium of the pulmonary system, has a lot of cilia on its surface. Okay? So these three are special examples of classifications: stratified, squamous, transitional, and pseudo-stratified. You should learn those because you will be examined on one or more of them at some point. Okay? Learn to distinguish these guys from simple or stratified columnar, simple columnar, whatever that we discussed previously. Here's some examples. Stratified squamous. So this is integument, skin of internal surfaces, like say in the mouth. You can see this layer of cells. There's a bunch of them. And here's the basement membrane down here. So there's a stratification of cells between the two arrows. And the uppermost layer of cells are squamous, i.e. elongated. So then this particular tissue would be classified as stratified, many layers of cells. Squamous, the uppermost layer of cells, is elongated. Okay? This is skin now on the surface of the body because it's got dead keratinocytes, but again it's stratified squamous because the uppermost layer of cells are squamous. We've got more than one layer of cells, several layers, and the uppermost layer of cells are squamous. So stratified squamous, in this case keratinized. And we'll learn more about this in greater detail when we do the integument, the skin and the various appendages of the skin. Here's another example of stratified squamous keratinized. Okay, again, this is the epidermis. You can see multiple layers of cells. There's only one basement membrane down here. There's no intervening basement membrane here. And all of these layers of cells are now becoming squamous. So stratified squamous, keratinized. Here's an example of stratified cuboidal. Okay, there's two layers of cells here. Here's the basement membrane, one layer of cells, because we're looking at the nuclei, a second layer of cells. So it only takes two layers of cells to make it stratified. Two's the minimum. And the uppermost layer of cells, furthest away from the basement membrane, is cuboidal. Okay? I think we have a computer brain freeze. Not sure what this is about. Let's take a break here. Is that the this thing? Let me turn it off.
It's working just fine, yeah.
Okay.
Do you want to hook this back up to you?
Yeah.
Carry on.
Yes, let's carry on. Okay, so we're recording, right? Yeah. Okay, so brief respite there. So let me see. Stratified cuboidal. So this is an example of stratified cuboidal because again there's two layers of cells, one, two, and the layer of cells furthest away from the basement membrane is more or less cuboidal. Okay, So this is stratified cuboidal, and this actually stratified cuboidal is pretty rare. The one example that we always give is the duct portion of the sweat glands, and this is it. So if you're looking at stratified cuboidal, you're probably looking at the duct portion of the sweat glands. That's usually how it goes. Okay, stratified columnar, okay, from the salivary gland. So here's the basement membrane here, bounding the perimeter of this epithelial sheet. Here's one layer of cells, based upon the nuclei, a second layer of cells. And the layer of cells furthest away from the basement membrane, these cells are columnar, okay, elongated. So this would be stratified, one layer, two layers, that makes it stratified. Columnar, shown here. Stratified columnar is pretty rare, okay? You don't see that often. You see it in the male urethra, you see it here in the salivary gland, a couple of other locations, not too many. Okay, pseudostratified. So here we have the respiratory epithelium lining the pulmonary system, the ducts leading down to the lungs and into the lungs. You can see here a bunch of nuclei that appear at different levels. Okay, one up here, one down here. So you would think, looking at this epithelial sheet, because here's the basement membrane that bounds the perimeter, but if you look at these cells, you're seeing nuclei at different levels, and you might think superficially that this is a stratified epithelium because you're seeing nuclei at different levels. But it turns out that every single one of these cells makes contact with the basement membrane. It might be in a different plane. So this one here, it doesn't look like it's making a contact here, but it's maybe like further into the image where it's making contact, okay? So this is pseudostratified, and as I said, pseudostratified is pretty rare. You'll come to recognize the trachea, or the pulmonary respiratory epithelium of the pulmonary system is one good example, and the epididymis of the male reproductive tract, epididymis vas deferens, is the other good example that we give. So if you learn those two locations and what they look like, then you'll be able to classify pseudostratified columnar epithelium. And obviously it's columnar because the uppermost layer of cells is columnar. And here, these also have cilia, which we would be able to resolve with an electron microscope. Okay, transitional epithelium. Remember I talked about transitional epithelium in the bladder, where depending upon whether the bladder is full or empty, the shape of those cells at the inside the bladder will change. So this would be actually an empty bladder because these cells are somewhat cuboidal in shape. The other thing that tells you that this is bladder is these binucleated cells. These binucleated cells are a distinguishing histological feature of the transitional epithelium lining the bladder. And that's an important phrase I just said there, distinguishing histological feature. What you want to learn for various tissues is something that is unique to that particular tissue that allows you to identify it as such and not something else. So in this transitional epithelium, binucleated cells will tell you that you are looking at transitional epithelium of the bladder. Okay. Firstly, you know it's stratified, but because you see these binucleated cells, you know that you're looking at the transitional epithelium of the bladder. So that would allow you to identify and classify this as transitional epithelium. Okay? Okay, so here's some different types of epithelium and their locations. So simple squamous, simple meaning one layer, squamous meaning elongated and flat. Okay, we talked about it. It can line various cavities of the body, okay, parts of the kidney, and also male reproductive tract, and in parts of the lung, pulmonary alveoli. Simple cuboidal, pretty rare. The best example, well, not so rare, pardon me, I was thinking of stratified cuboidal. Simple cuboidal, you see that again in a bunch of different glands and ducts as shown here. And we'll see all these as we go through the organs. Simple columnar, basically one layer of cells is columnar in shape. A lot of the GI tract, stomach, intestine, okay, parts of the gallbladder, and parts of the lungs as well can be simple columnar. Pseudostratified columnar is relatively rare. I talked about pseudostratified columnar being in the male reproductive tract, the epididymis vas deferens, the lungs, okay, trachea, bronchi, and also a little bit in the ear, the middle ear, the eustachian tube. The stratified squamous, so now we're looking at multiple layers of cells, stratified squamous. So the best example of that that's often given is the skin, okay, the epidermis of the skin, but it occurs in other locations as well. Stratified cuboidal, sweat glands, duct of the sweat glands is by far the best example. And we have some other transitional and germinal epithelium in the urinary system as shown here, which we'll discuss as we go along. Okay, so now we know how to classify epithelium based upon whether it's simple, one layer, okay, or stratified if it's more than one layer, and whether the shape of those layers, whether it's squamous, cuboidal, or columnar, and then there's some special examples, pseudostratified, transitional, okay, things like that. Now we can put those different layers of cells together to make glands. And we also classify glands based upon how those cells are put together. So the secretory part of the gland, we call that the parenchyma, the epithelial cells for the most part. And then these quite often are surrounded on the outside by connective tissue cells and extracellular matrix components, and we call that the stroma. So the secretory parts of the parenchyma, that's the epithelial cells. The stroma is usually the connective tissue around the parenchyma that kind of holds it in its shape. And there are different ways to classify glands. The first way is either exocrine or endocrine. Okay, exocrine or endocrine. So the way glands are made embryologically is we start out with our layer of epithelial cells, and you can see it starts to proliferate down to form the duct portion, and then from the end of the duct portion we'll get a glandular portion. And this would be an exocrine gland because now the secretions are going onto the surface. It can be an inner surface or an outer surface. And we can also have endocrine glands made the same way, but in this case they don't secrete onto a surface, they secrete into blood vessels. They can surround blood vessels, or sometimes like in the thyroid, they can secrete into a luminal cavity, which then can be transported to blood vessels that surround those epithelial cells. But in either case, the endocrine glands typically are secreting hormones of some variety into the vasculature. Endocrine glands are secreting secretory products, usually not hormones, onto a surface. It can be an inner surface or an outer surface, sweat glands onto the skin. So that's exocrine and endocrine. It's kind of easy to remember because endocrine, okay, blood vessels. If it's not endocrine, it's exocrine. That's it by default. Okay, so here's our endocrine glands again. Here's the thyroid follicle. You can see so the secretions go into this luminal structure, then it can be transported to blood vessels that surround the gland here. Or in this particular case, you can have these endocrine cells secreting directly into surrounding blood vessels. Okay, but in either case, endocrine glands are secreting usually hormones into the bloodstream, into the vasculature. Exocrine glands, on the other hand, possess both a duct and a secretory component. Okay, and again, these exocrine glands secrete onto the surface of the body or onto to surfaces within the body. So here's a generalized exocrine gland. You can see there's a duct portion which can branch multiple times, and then at the very end we have these small acina, which are the secretory portion. And if we magnify this little bulbous portion here like this, we can see the duct portion, which is the conducting portion in yellow, and the acinar portion, which in this case is the secretory portion, which is in orange. So the first way we classify exocrine glands is based upon whether the duct portion is branched or not branched. So the duct portion here is in pink. See, pink, 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 pink. If the duct portion is not branched, we say that's a simple gland, okay? If the duct portion in pink is branched, you can see a branch, more branches. If it's branched, then we say that particular gland is a compound gland. So that's the two distinctions based upon the structure of the duct portion. Not branched, simple. Branched, compound. That's it for the ducts. Okay? Then we can further describe and classify exocrine glands based upon the secretory portion. So yellow is the duct, right? So simple, not branched, compound because down here they're branched. But then we can describe the duct, the secretory portion. So if the secretory portion in orange is shaped like a tube, then it's tubular. Sometimes that tube can be branched, so that'd be a branched tubular. Sometimes that tube can be coiled, so that's a coiled tubular. And then besides tubular, we can have acini, which is like this grape-shaped structure. So we can see an acinus, single acinus, or in this case, a branched acinus. 1, 2, 3 branches, so that's a branched acina. And then we can have the same sort of arrangement of the tubular and acina secretory components in a compound gland. Remember, compound is a branched tube, and at the end of those tubes we can have a tubular portion. So this would be compound because the duct is branched, tubular because the secretory portion is tubular. We can have a compound acina because now the duct is branched and the secretory portion is acina. And sometimes we can get a mixture of tubular and acina. So here we have a compound tubular acina gland. Okay, so that's how we classify glands. On the duct, is it simple or compound? And on the secretory portion, is it tubular or acina? And if the tubular part or the acina part is branched, it's a branched tubular or a branched acina. And sometimes the tubular part can be straight or coiled. Okay? So here we have a compound gland because you can see that the duct portion is branched, right? It comes down here, 1 branch, 2 branch. And here we have different types of secretions. We have what we call a serous secretion and a mucus secretion. So let's talk about the mucus part first because it's probably easiest for you to comprehend. So mucus secretion, you know what mucus is. It's usually a light colored goop, yellowy light goop, not very dark, right? So that is typical of a mucus secreting cell. They're secreting these mucopolysaccharides, like it has a lot of sugar residues, okay, saccharides in there, which doesn't stain very well. So they typically stain quite lightly. So that would be a mucus secreting cell. In contrast, a serous secreting cell is secreting more aqueous or watery type substances. And because they're watery, they contain more proteins typically, and they stain darker. So a serous gland will stain darker than an adjacent mucus gland. So this is a compound tubuloacina. It's compound because the duct is branched. And there's a tubular component, which is the mucus component, and an acina component in this particular case because the serous cells are acina. Okay, then we can talk about mode of secretion. This is really talking about how an individual cell secretes products that it has synthesized for secretion. So perhaps the commonest mode of secretion is the merocrine mode of secretion, where secretory vesicles bud off from the Golgi and maybe have processing in them, and then those secretory vesicles will fuse with the apical plasma membrane and release those vesicles into the extracellular environment. So merocrine mode of secretion, most common. sometimes much of the apical cytoplasm will be secreted. So if you think of these small vesicles as the perforations of a stamp, if you had a whole bunch of perforations right across the cell, like you do with a stamp, a couple of stamps together, you can imagine if you've got multiple perforations, then that whole surface of the cell is going to rip off. That will be an apocrine mode of secretion. You see that in mammary glands a lot. There's a big chunk of the apical cytoplasm is secreted. That's the typical example. You don't see apocrine secretion so much. And then the third example of mode of secretion is holocrine secretion, where the whole cell, makes it easy to remember, holocrine secretion, the whole cell is secreted. So basically it's suicide. The whole cell is secreted and then it will rupture and release its contents. And a good example of the holocrine secreting tissue is the sebaceous glands of the skin, of the integument, okay? So merocrine is the most common. Apocrine, good example, is mammary glands. Holocrine, not so common, also a good example is sebaceous glands. For the most part, probably most of the tissues and organs that we're going to be discussing over the next couple of months will be secreting via a merocrine mode of secretion until we get to the mammary gland, until we get to the integument, where we'll talk about apocrine and holocrine as relatively unique examples. Okay, then I want to talk about cell surface specializations. I'm going to revisit these cell surface specializations on Wednesday when we're looking at cell organelles. So cell surface specializations are structures on the surface of cells like microvilli. Microvilli, cilia, stereocilia, and microplici. They're the four main types of cell surface specializations that you should be able to identify, okay, for exams. Okay, those four. So many cells, let's see here, did I go too far? No. Many cells are covered on their surface by microvilli. Okay, sometimes it's called a brush border because it looks like the bristles of a brush. But in fact, it's just a whole bunch of microvilli tightly packed together. So in this particular case, we can see the brush border where the arrow is flashing on the surface of an epithelial sheet from the intestine. And if we look at the same brush border with scanning electron microscopy, now we're looking at the surface, okay, of the brush, you can see the tips of these microvilli. These are all microvilli, and you can see why they would call it a brush border, okay? That's using scanning electron microscopy, so we're looking at the surface topography of the cell. And then with an electron microscope, we can actually look at a section through those microvilli. Okay, so you can see here, so here's the apical cytoplasm, the plasma membrane surrounding this one particular microvillus, okay, shown here. And we can make out a couple of different structures now in and around the microvilli. We can see inside the microvilli, and it contains actin filaments. And we'll talk a lot more about that on Wednesday. But that's characteristic of microvilli and stereocilia, actin filaments. And you can also make out this kind of fluffy, diffuse material on the surface of these microvilli, and this is what we call the glycocalyx. So this is a whole bunch of proteins and sugars that have been secreted by the cell that coat the cell. And this can be very thick, in fact. Okay, sometimes like, you know, a quarter of the diameter of a cell. So the glycocalyx is secretory material containing a lot of sugar residues that coat the surface of various epithelia. Now, if we take a cross section, if we cut a section this way through the microvilli, we can see now down into the microvilli. These are the multiple microvilli. And inside we can see now the actin filaments, these microfilaments within individual microvilli. Okay? So using it, and then of course here you can see this fuzzy layer around the perimeter of the microvilli. That's the glycocalyx of glycoproteins and sugar residues that have coated the surface of these cells. And in a schematic, we can see what this microvillus looks like. Again, we've got these actin filaments inside, which is characteristic of microvilli. And then it actually interdigitates with some other actin filaments running perpendicular just beneath the surface of the apical cytoplasm, which helps to anchor those actin filaments and allow those microvilli to be a little bit rigid. Now here we have cilia. Cilia are completely distinct from microvilli. They kind of look superficially like microvilli. They're on the surface of a cell. But you see how these are like thicker, have a thicker brushy appearance, right? Not as fun. The microvilli that we saw in the previous sections appeared fuzzy like a brush border. These are, you can see much more distinct striations. This is the respiratory epithelium of the pulmonary system. So right there, okay, it's kind of iffy if you can really say whether that's microvilli or cilia. But then when we start to look at that surface with different techniques, such as scanning electron microscopy, you can see that these cilia are much thicker and longer, okay? In this particular case, same epithelia. And now we know for sure that these are cilia, because when we look at the longitudinal section through these cilia, we can see a different structure inside. Inside these inside these surface structures, these cilia, we can see what we call an axoneme. And an axoneme is composed of microtubules. And these axonemes are also embedded into a basal body, which is a centriole. And the basal body nucleates the axoneme of the cilia. And if we look at a higher power view now, here's our cilia containing the axoneme. In cross-section, this is an axoneme. Here's a longitudinal section of an axoneme, compared with the microvilli. So MV is little bits and pieces of microvilli, that brush border that we saw of the intestine, next to much wider diameter cilia. And the cilia, you can see, have these long microtubules within them, distinctive microtubules that you don't see in the microvilli, okay? The microvilli are narrower. You don't see these long structures, which are the microtubules that we do see in the true cilia. Now, true cilia in cross-section, if they're motile cilia, contain a 9+2 arrangement of microtubules. So that is a unique, distinct histological feature of the axoneme, the 9+2 arrangement of microtubules. So you'll see that axoneme in true cilia. If it lacks the central pair, they're non-motile cilia, and you'll see that same structure in the tails of sperm. But basically, they're all composed of doublets of microtubules. So sometimes you can get defective cilia, like in the lungs. If you have defective cilia, you can't aspirate, you can't expectorate mucus that may collect in your lungs, like a lot of smokers may have issues with that, where they get defective cilia forming in the lungs after long-term smoking, okay? So here is a schematic of a motile cilia. You can see this doublet of microtubules and a central pair of microtubules, and these proteinaceous structures, the outer inner Dineen arms, and these radial spokes. So this is a schematic of what we hope to see in a high-resolution electron microscopic image of a true cilia. And that's what we see here. Not as clear as that, but that's what we see here. Doublets, 9 doublets in a central pair. We can have defective cilia. So in C, the inner Dineen arm, this little arm here, is absent, okay? So these cilia would be non-motile because those Dineen arms, they attach to the adjacent doublets and help to move the cilia. In D, the radial spokes are absent. These spokes here are absent. So again, defective cilia. In E, the central pair is missing, okay? So again, that would be defective cilia. And in F, both the inner and outer Dineen arms, the outer and inner arms, are missing from this particular cilia. So there are different ways you can get defective cilia. Okay, all of them then will lead to a lack of normal function of those cilia based upon what would be the normal structure of the cilia, which is the 9+2 arrangement of microtubules. So there's 9 doublets around the perimeter, and +2 is the central pair of microtubules in the cilia. So this is the typical structure that you will see for a cilia. You have to look for those doublets of microtubules. Okay? Whereas stereocilia, which we see in the epididymis in this particular example, have nothing to do with microtubules. The term stereocilia is a misnomer. Okay? These are really skinny microvilli. So do not confuse true cilia with the 9+2 arrangement of microtubules with stereocilia, which don't have any 9+2 arrangement of microtubules. They just have actin-like microvilli. So stereocilia should have been called stereomicrovilli. They're not cilia at all, okay? And you can see stereocilia, they're very long, skinny, microvillus-like structures. They kind of get entangled with each other from a scanning electron microscopic view. And in an electron microscopic view, you can see these very long, skinny stereocilia. There's no axonemal structures in those stereocilia. If you went to a higher power resolution, you would find actin within those stereocilia. So stereocilia, a misnomer. They're really skinny, long microvilli. They're not anything to do with the structure of true cilia. Microplici, they're the last thing that I want to talk about in terms of cell specializations. These are kind of surface folds, scanning electron microscopic view and transmission electron microscopic view. And you can imagine these little folds would help to retain mucus and various solutions that may be bathing over the surface of these epithelial cells. Like you see that in the GI tract in various other locations. So these are microplici. You don't see a lot written about microplici, but I'm mentioning them here because that's the fourth type of cell surface specialization. Okay? Okay, so much for epithelium and glands. Here's our question for today. Which of the following is not the operative word, not one of the 4 basic tissue types? Okay? Not one of the 4 basic tissue types. I see a lot of nodding, which means people think they know the answer. Do you know the answer? There you go. Glandular is not one of the 4 basic tissue types. It was epithelia, nervous, skeletal, connective, right? That's our 4 basic tissue types. Glandular is a combination of those things in some cases. Okay, so let's take a 5-minute break, if it's okay with you, and then we'll go on to the lab portion for today, and then we're done. We can just move through things, and then you'll have the afternoon off. Otherwise, we need to take an hour off for lunch and then come back. I assume you don't want to do that. You just want to continue on, right? 5 minutes. At 10 past, we shall resume. An eye on it, make sure I keep everything on. I'm good. We're good? Okay. Okay, so now for the third part of today's class, we're going to do a lab session, okay, so laboratory. So basically, this is a glorified show and tell. Like in the old days, when I first started teaching histology, we actually had a microscope, and we had a box of slides, and we would put that slide under the microscope, and we had a little booklet and said, you know, find this, look at that, all right? And you had to scan around and look at things, and you'd have professors and teaching assistants help you find things, which is actually a good way to learn, because it makes you really search for things and find things, and you can see things that look really good and things that don't look really good, but is the right structure. But in this day and age, we have reduced number of faculty and large classes, and we just can't do that anymore. So in its place, what we have is what we call a lab session, where I will show you, hopefully, a good image of a structure that otherwise you would have had to have found by yourself with the microscope, and we will discuss the characteristics of that structure, and you will have an opportunity to ask me questions about that structure. How do I know it's this and not that? What is this? What is that? Okay? So here I'm eliciting questions from you, more questions, okay? Because if you don't ask me any questions, I'm leaving in 5 minutes because we're done, okay? So, and it's up to you. And if you know it really well, great. Then we all get out, go have some lunch. Okay. So, and another thing I want to point out is, this is what would be on the lab exam. So remember, there's 2 lab exams. On the lab exam, you would be shown a structure, and there'll be a multiple choice question, A, B, C, D, E. Is it this, that, or the other? Or it may not be directly identification. It may be first you have to identify the structure, and then have to know its function. Okay. So it may be some gland that's secreting a hormone, and the question might be, which hormone does this particular cell type secrete? So first you have to know it's the thyroid gland before you could say that it's one of the thyroid hormones as opposed to some steroid hormone, testosterone, estrogen, whatever, okay, or pituitary hormone. So the questions can be varied in the way that they're structured, but you still have to be able to identify the structure to answer the question for the lab portion. Okay, so we talked about classification of epithelium first. Simple squamous. So in the schematic, it's simple because the cells, there's one layer of cells, right, on the basement membrane. The basement membrane bounds the perimeter in terms of classification. Here's our basement membrane. And there's one layer of cells above that basement membrane, so it's simple, and the shape of those cells is flat, so we call that squamous. Okay? That's pretty straightforward. Any questions about that one? Okay. And this is what simple squamous epithelium would look like looking down on, this is a piece of skin. So now you can see the polygonal kind of shape of the cells. You can see the nucleus and the cytoplasm. You wouldn't be able to say that this is either simple or squamous because we don't have a cross-section. We don't know that it's just one layer of cells, and we don't know that these cells are flat necessarily, but this is just giving you a view of that same tissue from a different perspective. Here again is simple squamous epithelium, actually endothelial cells lining a blood vessel. So here's the nucleus, a little bit of cytoplasm, and the basement membrane is just on the outside. So again, it's simple because there's one layer bounded by basement membrane, and it's squamous because they're flat. Okay? Is everyone okay with that? Yes? Not necessarily. You might be able to see it. I'd probably have a better picture than this one. But you'll know that that's a blood vessel. By the time you're done with this course, you can see in the middle here are all these red blood cells. Okay? Red blood cells don't have nuclei, the distinguishing feature of these particular cells. So you'll know that that's a blood vessel. So if that's a blood vessel, you know it's lined by a simple squamous layer of endothelial cells. The very first slide? Basement membrane is here, this kind of darker. Yeah. The basement membrane is always on the bottom of the layer. Oh, down here? This looks like a blood vessel. You can't make it out here. This is the nuclei and a fuzzy image of cytoplasm. You can't make it out because it's a low level of resolution for that slide. This one? Well, you don't really know it's cuboidal because we're not looking at a cross-section, we're looking at the surface. It could be cuboidal, it could be even columnar. We don't know because we're looking. So if my fingers are the cells, we're looking on the surface like this. So we don't know whether these cells back here, we don't know the shape, whether they're squamous, cuboidal, or columnar. Okay? Yes? This one? You can't really make it out, again, because the magnification is too low, so you really can't resolve the basement membrane. Yes? How do I know the arrow is smooth muscle? Because I know that this is a blood vessel. So based upon knowing what it is, I know that that's smooth muscle surrounding it. There are ways to stain for smooth muscle, specifically. It's not skeletal muscle and it's not cardiac muscle, so it has to be smooth muscle. And you'll be able to distinguish between those three types of muscle when Dr. Notaria gives you the structure of muscle. How do we know it's what? How do we know it's not stratified? I'm just giving you examples here. This is not necessarily something that you'd be examined on, but the way you would do that is by higher magnification. And then eventually, at a high magnification, you'd see a basal lamina and connective tissue, which bounds the basal perimeter of any epithelial sheet. So then you'd know there's just one layer of cells above that basal lamina, so therefore it's simple. And then when we look at the structure of blood vessels in the cardiovascular lecture, we also look at electron micrographs, so you get very familiar with the arrangement of blood vessels. The endothelial lining of blood vessels, it's always just one layer. You see this as a blood vessel. The endothelial cell lining is always one layer of cells, so it's got to be simple, right? And they're always elongated, so it's squamous. These things build upon themselves. That's why I also said, don't get behind in terms of learning the material as you go along. Review the lectures as you do them, as they get done. To kind of learn to be able to identify, classify things as you go along. And then, because that's basic, and you're going to build upon that. So then when we start putting a lot of these tissues together in terms of organs, I'll describe something as, a simple cuboidal epithelium in the kidney. Okay, and you should know what that means when I talk about simple cuboidal epithelium in the kidney. Okay, it kind of builds. But a lot of it also reinforces, because you'll hear the same terminology from lecture to lecture, and so it kind of reinforces itself, and you'll become, hopefully, familiar with these different classifications as we go along. Okay? So here our next example is simple cuboidal. So again, we look for the basement membrane or a different type of appearance. Here's a basement membrane here, and then the cells above the basement membrane are cube-shaped. So clearly it's a cuboidal epithelium, and there's one layer of cells, so it's simple. Okay, simple cuboidal. So then, if we look at an actual image from a histological section in this thyroid gland, you can see these cells are more or less cuboidal, and there's one layer. Okay, more or less cuboidal. So you have to use a little bit of judgment here. I mean, obviously there's going to be some point where cuboidal becomes squamous if you start to stretch it out, right? So you kind of have to use a little bit of judgment, because we could argue that these are actually a little more squamous than they are a little bit cuboidal. But then you look at a different section through the thyroid, and then they're going to look a little more columnar than they look a little more cuboidal. So it kind of can go one way or the other. But on the exam, you will be given very clear examples. The exam is not trying to trick you, the exam is trying to test you, okay? There's no trick questions. There's no trick exam questions. If there's a typo on the exam question, it's a typo, it's not a trick question, okay? So, we're trying to test, not trick. So that's a simple cuboidal epithelium. Okay, and then we have simple columnar epithelium. again, simple because it's one layer of cells resting on a basement membrane, and the cells are elongated, so clearly these are very columnar. And we have some different examples. This is very columnar. You can see how the height varies, though, even in this particular slide. Here's columnar. And these aren't quite as columnar, but they are more columnar than they are cuboidal. Okay? So again, simple columnar epithelial cells for classification resting on a basement membrane. Okay, any questions? Okay. And again, simple columnar, okay? I think this is a good example. Simple columnar, there's one layer of cells as identified by the layers of nuclei resting on a basement membrane. So simple columnar epithelium. Okay, and then many of these layers, like here, here, or other locations, may have on them microvilli. So the microvilli at a very high power image appear thus, and you can see very, very thin strands inside of them, which are the actin filaments. They're not microtubules. The microtubules will form the 9+2 arrangement of microtubules that you see in an axoneme of a true cilia. Okay, so this is not a cilia because we're not seeing an axonemal structure of 9+2 microtubules. We're just seeing very long, skinny actin filaments within these microvilli. And then also not very long cilia are going to be quite long because they're quite often motile, helping to move things along in a duct or some structure, right? So they're pretty much long. These aren't long enough to be doing that sort of thing. Also, you see this thick glycocalyx, so that's going to kind of glue everything together, right? So what's the point of this being motile if all of it's covered with goop and it's stuck together, right? You want a single cilia if you're going to be moving things to push things along. If you've got goop all over it, it can't be pushing things along. So again, the presence of this glycocalyx on the surface of these microvilli is also suggestive of these structures being microvilli as opposed to true cilia. Questions? Okay, so that was simple classifications, single layer of cells. Then we can have multiple layers of cells. So then if it's a multiple layer, it is a stratified epithelium. So here's the basement membrane, and we have 1, 2, 3, 4, 5 or 6 layers of cells, okay, with no intervening basement membrane. So multiple layers of cells, so therefore it's stratified. And the uppermost layer of cells is, in this case, flat, squamous. So this is stratified squamous, right? It's not stratified columnar, because down here against basement membrane, these cells are columnar. We don't care about those cells for classification purposes. We only care about this uppermost layer of cells furthest away from the basement membrane. That's the layer of cells that we use for classification. Stratified squamous epithelium. Here's some examples. Pardon me, this is the cornea. You can see these are more or less cuboidal, and then those layers at the in contact with the air are squamous, so stratified squamous. This looks like integument, skin. Again, stratified squamous. This is not a good example, so don't worry about that. Okay, stratified squamous again. This is the integument, the skin, the epidermis. This would be an internal cavity because there's no keratinized cells, but you can see multiple layers of cells, and then these are very squamous. So stratified squamous epithelium. Yes? Well, location, non-keratinized is internal cavity, so the lining of the oral cavity, your mouth, urogenital system, where it's just internal, typically is lined by non-keratinized. And histologically, non-keratinized epithelium does not have those dead keratinized squames. So on the surface of the skin, the surface layer of cells are dead keratinocytes, which is keratinized. And I'm going to show you an image. You can see histologically what they look like. It's the same basic tissue, but one is internal, one is external. So one is non-keratinized, the other is keratinized. So here's keratinized epithelium. Here's the keratin. So all of these keratinocytes have been flattened on the surface of the skin. They're all dead. Has a pretty unique appearance. Thin skin, it's not so thick. Thick skin, it's quite thick, as the names imply. So because we do have these dead keratinocytes, then it's keratinized, and the location has to be on the surface of the body. There is no keratinized integument inside the body. It's only on the outside of the body. Okay? Questions? Yes? Yes, they move up. So these cells down here are mitotic, and they become mitotic, and then they slowly move up, and then when they get to this level they die and become the keratinized dead squames that we see there. I will go over that in great detail when I do the integument lecture. I think it must be next month. Other questions? Okay, so stratified cuboidal. So sweat gland. So stratified, there's 2 layers of cells here. You can see 2 layers of nuclei. That's the minimum we need for it to be stratified. And the cells are more or less cuboidal. The uppermost layer of cells is more or less cuboidal, right? More or less cuboidal, depending upon where you look. It's going to vary a little bit, but the duct portion of the American sweat glands, that's the typical one we give as an example of stratified cuboidal epithelium. Any questions? Okay, so now some unusual examples of classification of epithelia. Pseudostratified, falsely stratified. Falsely stratified because every one of these cells in this epithelial sheet makes contact with the basement membrane somewhere. And there are no other cells sitting on top of other cells. Some of them pinch out, okay, here, or here, here and here. But every one of these cells makes contact with the basement membrane. But because we have different cell types within this epithelium, sometimes the location of the nuclei gives the appearance of it being stratified, because this one's higher than this one down here. So it's falsely stratified, okay? So that's a pseudostratified, and most of the cells are columnar, so then it's a pseudostratified columnar epithelium. The best 2 examples are the respiratory epithelium of the pulmonary system, which is shown here, and the epididymis and vas deferens of the male reproductive tract. They're the 2 examples you'll probably see for pseudostratified columnar epithelium. Questions? So here we can see pseudostratified columnar epithelium, respiratory epithelium. You can see nuclei, different locations, right? You would think, and here's the basement membrane down here. Looking at these, you would think, well, geez, this looks like it's stratified to me, okay? But hopefully, you would be able to recognize, it's got these cilia on top because these are much more fibrous than if they were microvilli, that this might be from the respiratory epithelium of the pulmonary system, in which case it would be pseudostratified columnar epithelium. So the question might be, classify this epithelium from the bronchiole, which is a component of the pulmonary system. So right there, I've told you the answer because I've said it's part of the pulmonary system. It's in the lungs, right? And when we do the lung lecture, you'll know the structure of all the different layers within the lung. So I've told you it's in the pulmonary system. So right there, And it looks stratified or pseudostratified, but you know, because I've told you it's part of a bronchiole or a bronchus, whatever, in the lungs, you'll know that that is the respiratory epithelium, and therefore it has to be pseudostratified epithelium, okay? I wouldn't show you that and just ask you to classify it, because it's not that easy to determine whether that is truly stratified or pseudostratified. But if I give it to you in the context of being in the pulmonary system, a bronchiole, then that nails it down for you, okay? So then, you know, sometimes you've got to put things together in the question. Then the question might be, you know, classify. So pseudostratified, stratified, columnar, pseudostratified, cuboidal, squamous, and so on, right? Transitional, whatever. You know it's pseudostratified. Any questions? Okay, and then here's some surface specializations. Our cilia again. Okay, here's our true cilia being nucleated from a basal body, which is a centriole. Centriole has actually triplets of microtubules, and we'll see that on Wednesday. That gives rise to these doublets of microtubules, which form then the cilia. And in cross-section, of course, we see a really good section of a cilia, the 9+2 arrangement of microtubules, which is that distinct histological feature of a cilia, which plays a role in motility of cilia. And you can see these true cilia. You can kind of see longitudinal sections through these microtubules here, next to a microvillus, which is filled with actin, which doesn't really show up anywhere near as clearly as the microtubules of the true cilia. Okay? So the fact that you can see microvilli adjacent to cilia helps you to identify one or the other in comparison, because you kind of know what's in the middle of these guys. Okay? Any questions? Okay, then on the surface, the cell surface, again we're looking at respiratory epithelium. You can see these true cilia that cover the respiratory epithelium of many parts of the pulmonary system. So it's just giving you a look at the appearance of true cilia with a scanning electron micrograph. This is wrong. This should say SEM. I have to correct that. Okay, that's SEM, scanning electron micrograph, not TEM. Any questions? Okay. Stereocilia. Okay, don't see it very often. Epididymis is a good location of the male reproductive tract. This is a low power view of the stereocilia. You can see how long they are. Basically, they're long, skinny microvilli, and because they're so long and skinny, they tend to goop together. So you see like little tufts of them, sometimes like little tufts. That's probably 20 different stereocilia there that have gooped together. And then if you look at an electron micrograph of our stereocilia, remember these are skinny microvilli. We don't see 9+2 axonemal structures, right? So we can say it doesn't look like a true cilia. Can't see anything because it's basically filled with actin. And this is a giveaway. What are these? These are sperm. You'll come to be able to identify the structure of a sperm when we do the male reproductive tract. And then you'll know that if you are looking at a sperm, you're in the male reproductive tract, which means that the lining epithelium is the epididymal epithelium, which is lined by stereocilia. Okay? So again, we looked at something else over here, we recognize it, that tells us what the organ is, and then because we know what this organ is, we can identify these as stereocilia because of what we know about the structure of the epididymis. So sometimes you have to look around and look at other stuff to be able to identify a particular tissue to answer the question about something else, okay? So take the whole thing in context. Any questions? You know, this might seem a little bit daunting now, but as we go along, this will kind of become second nature to you. It'll kind of build upon itself, and you'll really get a much better feel for identification and classification. Like as we get into the organ systems, we're going to look at very specific structures that make up an organ and how they work, and you're going to be able to classify those epithelia relatively easily, just because you've seen these different structures over and over again, and you compare and contrast to other ones that aren't quite the same. Transitional epithelium. Transitional epithelium, we're talking about the bladder, most likely the bladder, okay? It is stratified, but we don't describe it as stratified because the shape of the uppermost layer of cells can change depending upon whether the bladder is full or empty. So whether that upper layer of cells is squamous in shape when the bladder is full because it's distended, or that upper layer of cells is more cuboidal because the bladder is empty and so it bunches the cells up, right? So then that layer of cells is transitional in shape. It can be squamous or more or less cuboidal. So then we describe that as transitional. And in the bladder, sometimes we see, not shown here, binucleated cells in that uppermost layer. That's the giveaway. In the bladder, that's a histologically unique identifying feature that tells you that that is bladder epithelium and not something else, okay? So you want to look for unique identifying histological features that tell you that it's this and not that, and that's going to make your life so much easier, okay? So that's transitional epithelium, most likely the bladder. And here you can see empty and full bladder. This layer of cells is collapsed upon itself, okay? Whereas here they're distended. But in both cases, this is transitional epithelium. And I might pose the question, in the bladder, classify this epithelium. Simple squamous, simple cuboidal, stratified squamous, stratified cuboidal, and actually, and I might say, give the best answer, because one answer could be stratified cuboidal, perhaps, because it is stratified, and in this particular case it's cuboidal. But in the bladder, we always talk about transitional epithelium, so a better answer would be transitional epithelium. So, and actually right on the exam, the very first page of the exam, give the one best answer, because some of the other answers, although we don't try and trick you, might be a little bit confusing if you don't know the material very well, and there may be alternatives that really aren't the best. So you want the one best answer, okay? And in this case, because I've said it's bladder, the best answer is transitional epithelium. Questions? Yes? The answer, if I asked you to classify this epithelium in the bladder, the answer somewhere in there would be transitional epithelium. That's the answer. Okay, but the other alternatives might be simple squamous, simple cuboidal, stratified. Correct. Most likely. Most likely. The question has to be clear. Has to be unambiguous, because if it's ambiguous, you're going to complain to me. And I don't like gripes because it gives me headaches, right? So I try and make the questions as unambiguous as possible. There's one best answer, and that's going to be the answer. There was another question there first. So again, the top layer changes in shape. Well, actually all the cells, but it's usually the top layer of cells that we're looking at to see whether they've changed shape or not, right? So, pardon? Sometimes there is a binucleated cell not shown here, but I do believe, I know I have examples of binucleated cells of the bladder in my lectures, and we will see them at some point, okay? As a good example. And if you look at the text online, I think in Junckera, they show a picture of binucleated cells of bladder, transitional epithelium. Yes, that's the only, it's possible they're a little bit lower down, but I've only ever seen them on top. There's always that rare example where it's pops up where it's not supposed to be. But for 99% of the cases, it's going to be on top. Question in the back. Yeah. Yes, I might show you binucleated cells, yeah, and then say classify the epithelium. So you should know binucleated cells is a distinguishing histological characteristic feature of the transitional epithelium of the bladder, right? So yeah, that's another thing that could be used. Okay, other questions? Okay. Okay, so here's more bladder. Collapsed. Still not seeing these binucleated cells. Jeez, I have to throw one in there somewhere one of these days. I know I have a good example of one somewhere. Might be in Wednesday's lecture. But you can see here how these cells are really bunched up now in this relaxed bladder. Okay. Okay. Glands. So a single cell can be a gland, like these goblet cells of the intestine. They're secreting material, so we consider that a unicellular gland, single cell gland. And this is simple columnar, obviously. Goblet cell of the small intestine. And these goblet cells are secreting mucous material. They've got these big vesicles in the cytoplasm secreting into the lumen of the intestine, okay? And the secretion is a American mode of secretion. Most cells secrete by American mode of secretion, those vesicles that bud from the apical cytoplasm. Okay? Apicrine secretion and holocrine secretion, very rare. Apicrine in mammary gland, holocrine sebaceous glands, maybe a few other locations, but that's pretty much it. So everything else is going to be more or less American. Okay, by default, you can almost give that answer. Any questions? Okay, here's the same goblet cell in an electron micrograph. So here's the basement membrane. You can see these microvilli here on the apical surface, and here are these secretory granules being secreted from the apical cytoplasm into the luminal space here. So this is a granule here, or a vesicle. Okay, questions? Okay, classification of glands. Here's the duct, here's the gland. The duct is not branched. Not branched, so that means right away we say it is a simple gland. It's simple, right? And even though it's not that obvious in this particular H&E section, the glandular portion is a tube. Pardon me. So this would be a simple tubular gland in this particular case. And again, they're going to secrete by American because pretty much by default, if we exclude apicrine and holocrine and know where they secrete, which is pretty rare, most things are going to be American mode. Okay, so here again, the tubular part of this gland is simple. This is the tube of this sweat gland. Even though it looks like multiple cross sections, remember I talked about think in 3D, you've got a tube, you cut through it, it looks like there's multiple pieces, but it's not. It can be just the one duct. So the duct portion is not branched, so that's simple. Here's the secretory portion. Again, it's highly coiled, but it is tubular. So again, it is simple tubular gland. And by the way, not related to this, if you look at these, if the duct portion of this sweat gland, the epithelium is stratified cuboidal. This is where we see stratified cuboidal pretty much all the time. Stratified cuboidal in the duct portion of the American sweat glands. That's the example we typically give because that's the best example we have to show. Okay, but again, in terms of glandular classification, it's simple because the duct is not branched and the secretory portion is tubular. Okay, simple tubular. Any questions? Yes. Not always easy. You need to get a good example. So I would show a good example where you can see the branch. And in this one, you would look at this and you would see that the duct portion is stratified cuboidal. So you know you're looking at the American sweat gland. So you know, because it's American sweat gland, it is simple coiled. All right? Simple coiled tubular. So by identification of this epithelium, it actually tells you what you're looking at, and then you remember, oh yeah, okay, sweat gland, American sweat gland. So the other things, like I said before, sometimes look for other stuff that can give you a hint as to what you're really looking at, and then that can clue you to the answer that you're looking for. American sweat gland. Okay, gastric glands. Here we have, again, it's simple because the duct portion is not branched, but now the tubular portion is branched. So this is simple, branched tubular. It's branched tubular. Okay? Simple, branched tubular gland. And again, it's American secretion because most things are. Questions? Okay, sebaceous gland. Simple because the duct is unbranched, although we can't see the duct here. And the acina portion, the secretory portion, is acina. So it's simple, branched acina. Here's the acina. And because this is a sebaceous gland, the entire cell is released as part of its secretory mechanism. So this is an example of holocrine secretion. The whole cell is secreted. Holocrine secretion. This is the best example of holocrine secretion, a sebaceous sweat gland. So if you can identify this as a sebaceous sweat gland, you would know that these cells are, the mode is holocrine secretion. In the skin, all over, pretty much all over, yeah. Other questions? Okay, maybe not so much on the internal surfaces, but the external surfaces. Okay, compound acina. So before everything we looked at before was simple. Now we're looking at compound. Compound means the duct portion is branched. Okay. Compound is not used in the context of the secretory portion. It's only used in the context of the duct portion. So here's our gland, and it branches, right? Multiple branches. So because the duct portion is branched, this is a compound gland. And then if we go further down and we look at the secretory portion, they're kind of bulbous, so they're saying this is an acina gland. So it's a compound acina gland. And if we look at the contents of the cells that are secreting, they stain rather darkly, so that's a serous secretory cell, not so much a mucus cell, because mucus is light colored and goopy. Serous is much more darker stained, so that's a serous gland. Okay, and again, it's a merocrine mode of secretion. Okay, now we're looking at the mammary gland. So the duct portion, branched, therefore it's compound. The glandular portion is acina, kind of grape shape, so it's a compound acina. And because it's the mammary gland, a big chunk of the apical cytoplasm is secreted as one big secretory vesicle, so then that's an apicrine mode of secretion. That's the example we give for an apicrine mode of secretion. And you can see the apicrine vesicles. It's a huge, it's almost the diameter of the apical cytoplasm being secreted as an apicrine secretory product, okay, in the mammary gland. Okay, and then sometimes we can have mixed mucus and serous together. So here in this submandibular gland, the duct is branched, okay? So that's compound. And then we have both a tubular component and an acina component. the glandular material is also mixed mucus, because the mucus material stains lighter. The darker stained material is typically serous. Okay, so that's a mixed serous mucus gland. And again, it's merocrine. Okay, and then here you can see the mixed merocrine and mucus secreting material. Here's the merocrine component, and here's the mucus component, so the serous component. So the serous is this, which is this. The mucus is this, lighter colored stained material, which is this, the big light colored secretory vesicles. Okay, so in the same acinus here, we've got mucus and serous secreting cells. Okay, questions? Yes? The orientation, yeah, it could be like cut through here. It does. The orientation is hard to say for sure because it's cut through everything. You've got the duct portion. So let's see here. It might be like maybe like cut through here, maybe, because these secretory components can be all over the place. It's just captured everything. It's out here somewhere. It's not up here where you've got the big duct. It's down here where we've got little duct. Okay. Other questions? Okay. And then sometimes, like in the pancreas, we can have both exocrine and endocrine glands. Remember, endocrine glands are secreting hormones into the vasculature somehow, such as in the islets of Langerhans, secreting insulin into these blood vessels. And we also have exocrine components outside of the islets of Langerhans, such as here, this serocrine secreting material in the other part of the pancreas. So this is a mixed exocrine and endocrine gland, the pancreas. Okay? Okay, merocrine mode. Okay, and here it is again, the acinus cells. We can see the secretory vesicles being released from the cells. Okay, that is it. So here's our question, last question. This lining epithelium is classified as pseudostratified columnar, transitional, simple columnar, stratified columnar, stratified cuboidal. Okay, look at the layer of cells. So we're talking about from here to here. Okay? This layer of cells from here to here. Okay, well, it's not simple, is it? Definitely not simple. It's columnar of some description. That narrows it down. Okay? People look a bit confused. Is this tough? Okay? Do you think you know the answer? Simple columnar. Does anybody want to ask me why it isn't one of the other ones? Why it isn't something else? Yes? You need to speak up. They're microvilli. It's not pseudostratified because it's not epididymis and it's not respiratory epithelium. Respiratory epithelium is going to have true cilia, very long. Epididymis would have sperm out here. Okay, these are microvilli, the brush border, and really the cells, the nuclei, are pretty much arranged in one plane. I'm not seeing any basal cells down here, which you will see in pseudostratified. Okay, it's pretty much a uniform layer of nuclei in the same plane. So it is really simple. Okay. Other questions? Yes.
You said that
stratified columna in the male urethra, the penile urethra of the male, not in the female, only in the male. That's the other common location, stratified columna. Other questions? Okay, there you go, guys. So that's it for our introductory series of lectures. And then I will see you on Wednesday when we're going to do a deep dive into all of the cell organelles and junctions between cells and some cell surface specializations again in detail. Okay.